DNA replicates itself, and then what its function is, so what its job is in the human body. So your book starts out by talking about experiments done by Avery and McCarthy, and then also Griffith. So in the early 1900s, Griffith, what he discovered is that if he had some mice, you guys can read about that right here. I'm just going to show you the picture here real quick. What he did was he injected mice with a strain of bacteria called the rough strain. That's where this R comes from. And when he did that, the mice lived. They were fine. But when he injected mice with a different strain, a smooth strain, it killed the mice, made them sick and die. So what he did was he attenuated the bacteria, or he kind of ground it up and broke it so that it wasn't working properly, and he injected mice with it, and they didn't die, they were fine. But when he injected mice with the rough strain, which in the previous experiment over here didn't do anything, the mice were totally healthy, and then he also injected the mice with a combination of the rough broken strain, so the one that had been attenuated or killed, the mice died. So kind of going off of that, what they found is that DNA or that bacteria can kind of trade notes, and so that non-virulent rough strain actually was able to gather something, we don't know what it is yet, but they were able to gather something from that broken rough strain, or that broken smooth strain, and then turn into the deadly type of bacteria that killed the mice. So kind of building on that experiment, Avery and McCarthy, what they did was they separated that smooth strain into separate parts, and so they separated it into lipid parts, protein parts, and then the nucleic acid or the DNA components. And then what they did was they took those and they put them also into mice, and they found that the DNA was what was called the transforming principle, or what was actually being used by the bacteria to grow into the virulent strain. And so later on in the 1950s, there was a couple called Hershey and Chase, and what they did was they took a bacteriophage, and that's just a big fancy term for a virus that infects a bacteria. They kind of look like a little spaceship. I'll show you a picture here in just a second. But what they did was they labeled different parts of that bacteriophage, and they looked at where those parts went, and so they used what's a 35 sulfur, and then they also used a 32 phosphorus. And the important part here being that the sulfur, its job just kind of like was to hang on to the outside protein part of the virus. It's not used, and it wasn't taken into the DNA part. But phosphorus, if you guys have looked at what makes up DNA, phosphorus is a big part of DNA. So the phosphorus was actually taken up by the bacteriophages, and they were injected into the bacteria, and that phosphorus became part of the DNA, and so it was like radio tagged, so they were able to find out where that radio tagged phosphorus went. And what they found is that the phosphorus actually did attach to the DNA, and the DNA was what was being passed from the bacteriophage to the bacteria. And so that kind of looked like this. And so there's a bacteriophage for you. It looks like a little spaceship. Here's a scanning electron micrograph of what they look like. Kind of creepy little spaceship dudes. But this is the protein coat right here, this part that looks like the big spaceship, and then you can see the DNA just kind of wadded up inside of the capsule right there. And so what they found was that when they labeled with the sulfur, it just attached to the capsule, and that was never found in the bacteria. So even after the bacteriophage injected its DNA and took off, Hershey and Chase looked at the contents of the bacteria, and they didn't find any traces of that 35 sulfur. So they were able to see that that was attached to the protein. It wasn't used, and it wasn't transferred to the bacteria. However, down here with the phosphorus, and again, remember, phosphorus is a big part of DNA, and so that's how they were able to attach this to the DNA, is they were able to get the bacteriophages to pick that up and then uptake it so that it could be used to be part of their DNA. And so when they bombarded these bacterial cells with these bacteriophages, these little spaceship guys right here, they were able to find that radio-tagged phosphorus inside of the bacteria. So from this, they were able to deduce that DNA is the hereditary portion of the bacteriophage and of the bacteria, because that's what's basically passing the notes around. So here we go with our bacteriophage injecting the DNA, and now we're able to pick up traces of that phosphorus here, and that's how they were able to kind of figure out that DNA was the hereditary material, because that's what's being passed in between the bacteriophages and the bacteria. So later on, we started getting some information from different experimenters and different scientists about what DNA structure actually looks like. You guys probably remember Rosalind Franklin and then Watson and Crick with their famous experiments on DNA structure. We'll talk about them more in a minute. But what we discovered about DNA was that DNA is made out of monomers. Remember, just like the little single parts of DNA, the little single pearls on a string of pearls, if you will. The monomers of a DNA are called a nucleotide. And so a nucleotide is made of a sugar, and if it's DNA, that sugar is called deoxyribose. Phosphate groups, and so that's where the phosphate from those Hershey and Chase experiments were able to integrate into the DNA. And then one of 4 nitrogen-containing bases. And so this is where the actual information is coded, is in these nitrogen-containing bases. And so the nitrogen-containing bases are of 2 different structures. There's either pyrimidines or purines. And so pyrimidines are a single ring structure, and then purines are a 2-ring structure. And I'll show you what that looks like here in a second. So the 2 pyrimidines are thiamine and cytosine. The way that you can remember that are that the pyramids are in Cairo and Thebes, which is a total dirty, rotten lie. I don't think that they're actually located in Cairo and Thebes, but if that helps you remember that thiamine and cytosine are the pyrimidines, then go ahead and use that mnemonic. And the purines are adenine and guanine. And so the way that these look, so you can see here we've got our cytosine and thiamine, so these are our 1-ring structures. These are the pyrimidines. So remember, the pyramids are in Cairo and Thebes. And then we've got our purine structures, which is a 2-ring structure. So we've got these little guys right here, adenine and guanine. Now, those, the way that they base pair up on a double-stranded DNA strand is always, adenines always pair with thiamines, and guanines always pair with cytosines. And so Chargaff's rule just states that the amount of adenine is going to be about the same as the amount of thiamine. In fact, it should be almost exactly the same. And guanine is going to equal the amount of cytosine, because these 2 always pair together, and so it kind of goes without saying that if you have 40% guanine, you should have 40% cytosine, because they always pair together. And so the proportion of this adenine and guanine, so of the pyrimidines and the purines, is going to vary a little bit, because it doesn't matter if you have 40% adenine, that means you're going to have 60% guanine, because we have to add up to 100%. And so the amount of pyrimidines and purines that we have, that ratio might be a little different, but the amount of A's and T's should always be the same, and the amount of G's and C's should always be the same. So Franklin, Rosalind Franklin, and then Watson and Crick, they did some really interesting research using what's called X-ray crystallography, and so they were actually able to crystallize DNA and then take X-ray pictures of it, and those pictures were interpreted by Watson and Crick to be a double helix. And so these guys were actually responsible for figuring out that DNA was a double helix, because before this we just knew that it was the hereditary material. We knew that it was made of phosphorus and a sugar and some bases, but we didn't actually know what the structure actually looked like. And so these guys together with Rosalind Franklin's X-ray crystallography, and then after being interpreted by Watson and Crick to be a double helix, they were able to figure out what DNA looked like physically in real space. And so that picture is a double helix, and I'll show you guys pictures of that here in a second. And so a DNA molecule is what's known as double-stranded, and so DNA molecules consist of 2 nucleotide chains, and so one, it looks kind of like a ladder going up and down, and so you've got one side of the ladder that's got the side and then half of the rungs or the steps that you would step on, and then the other half is exactly the same. So you've got the 2 sides, and then you've got the rungs of the ladder in the middle. So the base pairs form the inside of the ladder, and so those A's and T's and then the C's and G's form the inside of the ladder that you would step on if you were looking at this as a ladder. Hold on just a second. Oh, there we go. So this is what that double-stranded structure looks like. So here we go right over here. You can see this very tiny 0.34 nanometers between each base pair, and so that's between this guy and this guy, 2 nanometers in diameter, so that's 2 billionths of a meter. Remember, tiny, tiny, tiny little measurements here. And then also 3.4 nanometers for each full turn of DNA here, so 1 full twist of that helix. So eukaryotic chromosomes and then also prokaryotic DNA look a little different. So eukaryotic DNA is organized into chromosomes. You guys may be familiar with that terminology. We've got, as humans, 46 single chromosomes or 23 pairs of chromosomes, and so our DNA is organized into like singular chromosomes. In prokaryotes, they have a ring of DNA, and they just will split off that one ring, and so it's like this big spaghetti mess looking kind of mess of DNA. Eukaryotic chromosomes are a little bit more organized. So we are organized into those chromosomes, and they are, each chromosome is different in its length and shape because they carry different genes on them, and so they're all different lengths, which means that they're different sizes. So a chromosome is just a structure that consists of DNA that's associated with proteins. What they mean by that is that it's wrapped around these little spools, if you will, kind of like thread being wrapped around a spool, so that way it doesn't get all twisted up. If you guys have ever taken like a length of rope or something and you've just thrown it into your garage, and then you go out to get, oh, Christmas lights is a good example of this too. So if you've ever taken Christmas lights, okay, and you just kind of like jumble them up and throw them into a big bin, and then you take them out next year and they're just like a huge jumbled Christmas light, like horrible mess the next year. You've probably all like experienced this in one form or fashion, whether it's with yarn or Christmas lights or thread or something like that. You know that it's much easier to unravel Christmas lights if you wrap them around a spool, or if you wrap thread around a spool, or if you wrap yarn into a like a center pole ball or something like that. And so DNA is kind of the same thing. We want to wrap it up so that it's very easy to unravel, and we want to wrap it up so that it doesn't get all tangled up. And so that's what those associated proteins are for. They're literally like the spool on a spool of thread. And each chromosome is going to carry a part of the genetic information for that organism. So during most of the cell's life, okay, so like when it's just doing its thing, when it's being a bone cell, when it's being a nerve cell, when it's being a skin cell or whatever, the chromosome consists of just one DNA strand. You only really see the chromosomes condense down during cell replication. So when the cell is preparing to divide, it's going to duplicate all of its chromosomes so that way it can separate one set of chromosomes into one of the offspring and one set of chromosomes into the other, so that way each new resultant cell will have a full set of chromosomes. Then it's going to duplicate all of its chromosomes. That way it can split them in half. So each duplicated chromosomes, or each duplicated chromosome is going to look kind of like an X. Whenever you see a chromosome that looks like an X, it has been duplicated. And so this is what that looks like here. So this is one chromosome unduplicated. So this is what a chromosome actually looks like. It's just like a single filament. And if we've duplicated it, preparing for cell division, it's going to look like this X here. Okay? And so when we separate the two cells that we're going to be making when we do our cell division, these are going to get pulled apart. And so the centromere is what's holding them together at this moment. But we're going to pull them apart. We're going to put them in separate cells, and then each cell will have a full set of chromosomes, which is important. Especially in humans, we don't do very well if we don't have the right number of chromosomes. So, like I was talking about before, each coil of DNA is wrapped around those spools, and those proteins that act like those little spools of thread are called histones. And so histones come together and they form a group of 8, and that's called a nucleosome. So it's almost kind of like putting several spools together and then being able to use them as like one super spool. And so this is the smallest unit of chromosomal organization in a eukaryote. Okay, so all we're doing here with our DNA is we're wrapping it up because we don't want it to get tangled and we don't want it to turn into the huge Christmas light mess. That would be bad. We don't want that to happen. And so very carefully, your body is going to wrap your DNA around these histone units, and then we're going to put those together into kind of a bigger nucleosome unit. And then we're going to supercoil them on top of each other until that DNA is what's called super condensed. So the super condensed DNA is going to be visible for you under a light microscope because it's so tightly wound on itself, and it's also going to be used for duplication and replication. So that's what this looks like. So looking here, we've got our single strand of DNA. So we've got our one side here. This is your sugar base. This is your phosphate right here, and then you've got your A's and C's and G's and T's. So your nucleic acid bases in here. Okay, and so these are then kind of helixed up, and so those turn into like what's called a one coil kind of configuration. Then we're going to start wrapping this DNA around these little histones right here, these little proteins, because again, we don't want them to get twisted up, we don't want them to get turned into the Christmas light debacle of 2001 or whatever. And so we're going to wrap these little guys up, and then we're going to start supercoiling them. So you see how organized this is, and so we've got our little wraps of DNA around these histones, and then we're going to supercoil them together. And then this mass is also going to supercoil again in and on itself, and then we're going to coil it again. Okay, so this is where that supercoiling kind of takes place. And then once we've got all this supercoiled and super condensed, we can turn them into chromosomes. And so here's our big chromosome right here. And now this structure, the number 5 structure here, this is actually visible under a light microscope. So when you guys look at these in lab, you're going to be able to see them very easily with a light microscope under about 40 times magnification or 400 total magnification, pretty easily. And that's because the DNA is so super condensed that you can see it under a light microscope. If it were just in this configuration, it would be so thin that you couldn't see it. And so that's the important thing to recognize here, is that this DNA is so organized and so super coiled on itself, kind of like a phone cord, one of those old school phone cords, just wrapping and wrapping and wrapping and continuing to wrap on itself until it just turns into a big super coiled mass, and then you can see it under the microscope. So here's what that looks like, and you can see that this is much easier to see, this big chromosome right here, and you can see all that DNA in there. That's kind of cool. Okay, so the total number of chromosomes in a eukaryotic cell, remember that this is in plants, animals, fungus, protists, those kind of things, the chromosome number is characteristic of the species. So you guys probably know that you have 46 single chromosomes, or 23 pairs of chromosomes. So you get one set from your mom, one set of 23 from your mom, one set of 23 from your dad, and that adds up to the 46 chromosomes. So there are 2 of each chromosome. So you get one chromosome number 1 from mom, one chromosome number 1 from dad, okay? And so you have 23 pairs of chromosomes. So our diploid chromosome number is 23 pairs. And so the diploid number is always denoted as 2n here, which means 2 sets. The n refers to sets. And so since you've got one set of chromosomes from your mom, one set of chromosomes from your dad, your diploid number is 23n, basically. And so the diploid number is called 2. That means you have 2 sets. And so looking at a picture of the chromosomes, we can see how many chromosomes an individual has, and we can see like if there's any abnormalities or if there's any extra chromosomes or if there's not enough chromosomes. So a picture of an individual's chromosomes is called a karyotype. And so this individual, you can see here, we've got 2 chromosome number 1s, one from mom, one from dad, 2 chromosome number 2s, one from mom, one from dad. And if you add these all up, that's going to equal 46. Now this individual is a girl. We can tell because she's got 2 X chromosomes right here, okay? If this individual were to have 1 X chromosome and 1 Y chromosome, that would make it, you guessed it, a boy! Oh my gosh, so exciting. And so this individual has got 2 X chromosomes, and so that makes her female. Boys have 1 X chromosome and 1 Y chromosome, and we'll look at more about why that is a little bit later. So in a diploid organism, so an organism that has 1 pair of chromosomes from mom, one set of chromosomes from mom, 1 set of chromosomes from dad, 1 pair is inherited from the mother, 1 pair is inherited from the father, okay? So all of the chromosomes, I'm going to go back to this picture here real quick, all the chromosomes from pairs 1 through 22 are known as autosomes. That means that they're the body chromosomes. And so all pairs 1 through 22 are homologous, meaning that they have the same genes on them. So chromosome number 1, for example, might have genes for height or weight or something on there. And so chromosome number 1 from mom is going to have height and weight genes on it. Chromosome number 1 from dad is also going to have height and weight genes on it. Chromosome number 2 might have genes for eye color. And so chromosome number 2 from mom is going to have eye color genes. Chromosome number 2 from dad is also going to have eye color genes. That's what I mean when I say that these chromosomes are homologous. They have the same genes on them. Now, when we start talking about genetic differences between people, you might have the same genes on the same chromosome, but it is possible to have what are called different alleles on the same chromosome. And an allele is just a different form of a gene. And I'll explain that here in a second. So if you've got eye color chromosomes, for example, so like we'll just pretend that chromosome number 2 holds all the genes for eye color, okay? Eye color is the gene, and then maybe blue eye color would be the allele. So everybody has genes for eye color, but you might have genes for blue and then a brown. Maybe your mom has blue eyes and your dad has brown eyes. And so you have one allele for eye color that's blue, one allele for eye color that's brown. And so the allelic part, the different form of the same gene is the color variation. Same thing for hair, same thing for skin tone. You end up with all these different combinations of genes, which are the different alleles, but everybody has genes for skin color. Everybody has genes for eye color. Everybody has genes for height. But the difference is that you might have the short height gene, or your husband or your wife or whatever might have the tall genes or whatever. And so that's the differences between the two. And so chromosome pairs numbers 1 through 22 are called autosomes, which means that they code for all the body stuff. Auto means body or self, and so these are all of your self chromosomes. Now, for a female, she's going to have homologous chromosomes, so chromosomes that all hold the same genes all the way down the line. This last pair down here is the 23rd pair of chromosomes, and they're called the sex chromosomes. And so for a female, all of her genes and all of her chromosomes are going to be homologous. For a male, or for someone who has an X and a Y, this last pair right here are going to be non-homologous, because X chromosome and Y chromosome do not have the same genes on them, okay? The Y chromosome doesn't have any essential genes on it, which is good, because if that were true, then all females would die, because none of us have a Y chromosome. So everybody has to have an X chromosome, because that chromosome does have essential genes on it, but not everybody has to have a Y. The Y chromosome has the genes for maleness on them, and so those are not essential to live, but they are important if you're a boy, for sure. That's what's going to give you all of your secondary sexual characteristics and even all of your primary sexual characteristics, like having a penis and testicles instead of ovaries and a vagina. And so it's important, especially if you're a boy, like I said, to have that Y chromosome. And so if you're a boy and you have one X chromosome and one Y chromosome, this last pair here are known as non-homologous. And if you're female, all of your chromosomes are homologous, because they've all got the same genes on them. So everybody, their pairs 1 through 22 are going to be homologous chromosomes. They've all got the same stuff on them. And if you're a boy, then that last pair is non-homologous. And again, that 23rd pair is called sex chromosomes, because they determine your biological sex. And pairs 1 through 22 are called the autosomes, because they are everybody has the same pairs 1 through 22, because they've all got essential genes on them for how to build a human. So you got to have all those. Okay. So the way the DNA replicates itself in order to make more cells, because every time we replicate our cells for growth or for repair or for whatever, we have to put a full set of DNA into each of those cells. So that way the cells themselves have a copy of all the recipes on how to make a human. That's what we need. And so we have to replicate our DNA, and we're constantly replicating it in almost all the cells in your body. Not all of your cells are going to undergo cell division, but most of them will. So we have to replicate our DNA in most of our cells. This is a really energy-intensive process. We have to copy all of our DNA. We have to make sure that there's no mistakes, because that's how cancer happens and that's how mutations happen. So you've got several checkpoints while your DNA is replicating itself, where you've got enzymes that will go in and actually check and make sure that this is happening properly. So a cell, before it thinks about replicating, is going to reproduce all of its DNA. It's going to copy all of its DNA and make 2 copies. That way, when we get ready to split, all of our DNA is already prepared. We've already made 2 copies of it. That way, when the cell splits, we can just split that DNA in half, and then each new resultant daughter cell is going to have a full copy of those chromosomes. So DNA replication, like I was talking about before, it has a lot of enzymes that are included in it and that are involved in making sure that this replication process takes place properly and with no mistakes, because we don't want to get a mutation or something crazy going on when we replicate our DNA. We want no mistakes to happen because we don't want to get cancer or a mutation or something crazy going on. So the cell's genetic information does consist of the order of the nucleotide bases. And so just like we were looking at before with the one side of the ladder, the order that those bases are in, so it might be like GATTACA or something like that, that's actually what conveys the information for how to make proteins. And so all of the cells that are coming from that parent cell need to get an exact copy of that information. If they don't, that means they're not going to make, they're not going to have the right recipes for how to make a protein. They're not going to have the right recipes for how to make a human. And so it's very, 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 I mean, like fatally important that we get an exact copy of this information. So each chromosome is copied 100% to its entirety. And the 2 chromosomes that we get as a result of this is going to be exact copies of one another. And again, it's very important that this is exactly the same. If we get too many crazy looking mutations in there, that can cause cancer, it can cause mutations, it can cause our proteins not to work properly, and in some cases that can be fatal. So we want to make sure that we have an exact copy of our DNA. So the first thing we have to do to get our DNA to duplicate is we have to unwind it, because again, like the order that these little bases are in right here is the keys to the kingdom. Okay, so we have to make sure that these little bases are in the right order. And so the first thing we have to do is we got to break or melt our DNA in half. And so there's a helicase enzyme that comes in and it breaks the hydrogen bonds that are holding the DNA together. If you guys remember from the first couple of chapters, we talked about hydrogen bonding is not crazy strong, but it is strong enough to keep your DNA together, and it is strong enough to keep the cohesive properties of water. And so helicase, his job is to go in there and melt the DNA or break those hydrogen bonds in between the 2 strands. And so that's what we're doing here. So here's your helicase right there, and we're going to melt those bonds between the DNA. You have another enzyme that comes in, it's called topoisomerase, and it's going to untwist that DNA double helix. And so we're going to unwind it and make sure that we're getting those 2 separate strands apart from each other. And then a polymerase enzyme is going to come in and it's going to join free nucleotides into a new strand of DNA. And so I'll show you kind of what that looks like here in a second. So all we're doing is a DNA polymerase enzyme is going to come down and it's going to read the order of these bases right here, and it's going to add bases on the other side that complement each of these bases. And again, I'll show you what that looks like here in a second. So polymerase's job is to, like just like the polymer right here would imply, this DNA polymerase is going to make a new polymer of DNA. And so it's going to do the complementary base pairing for us, and it's going to literally copy one side of the DNA and the other side. Then, once we're done doing all of that, we've got DNA ligase comes in here, so this guy, and it's going to join DNA segments if they're discontinuous. And so if we need to rejoin strands of DNA together because they've been broken or whatever, ligase is going to come in there and do that for us. So there are several different types of DNA polymerases. In our case, we're going to talk mostly about DNA polymerase 1 and 3. So there's lots of different types of DNA polymerases, and remember that DNA polymerase, his job is to read and produce new polymers of DNA. So his job is to literally read the bases on the one strand of the DNA, and then to come in there and do the complementary base pairing for us. And so each of these requires a primer to initiate DNA synthesis. A primer is just kind of like the DNA's version of like a starting line. And so that's where the DNA polymerase knows that, like, okay, we're going to start copying here. It's kind of like a little flag. It's a little piece of DNA or RNA that says, like, woohoo, hello, I'm over here. Start the initiation of the polymerase over here. This is where you can sit down, and this is where you can start reading, basically. And so primer, again, is a short single strand of DNA or RNA, and it's basically like a big flag that says, like, hello, come over here, polymerase. This is where we're going to start replicating our DNA. And so DNA replication is known as what's called semi-conservative, because when we break our parent or the original strand of DNA in half and we build a new strand on the other side of it, each new strand of DNA is basically going to have one strand of old or parent DNA, and then the other side is going to be new, that was built by the DNA polymerase. And so each strand of DNA in a double helix is a template, so that's what we're building our other side on for the synthesis, so that we can make a complementary strand of DNA, so one that complements the other side. So one template builds DNA continuously, that's the 3 to 5 prime end, and then the other is going to build DNA discontinuously, and I'm going to show you what that looks like here in a second. And so each new DNA molecule has one strand of the old, or the parent DNA, and one strand of new that was just built by the DNA polymerase. And so again, this is called semi-conservative replication, because we are partially conserving the original DNA. We're not just totally chewing it up and, you know, making a brand new strand. We're using half of that old DNA in order to build a new strand of DNA on top of it. And so here we go. So this is our DNA replication. If it were me, I would look at these different starts here and all these different phases, and I would commit them to memory if it were me, just if it were me. And so what we've got over here is you've got your regular, this is our parent DNA. We've got our topoisomerase and our helicase enzymes, and they're going to unzip the DNA here. And so now you can see we've got two parts of DNA, or two sides, because we've melted, or we've destroyed those hydrogen bonds in between the two original strands of DNA. And then we've got our polymerase here. So our polymerase is going to come in here, and it's going to read this side of the DNA, and it's going to add base pairs. And so, like, if this were a C, for example, then the polymerase would be like, okay, I need a G over here, because remember, C's and G's always go together. And if this were an A, this would be a T. Okay, and so polymerase is going to read this side of the DNA, and it's going to add those complementary base pairs to the other side. And so what we end up with is two new strands of DNA that are made of half old DNA, this blue right here, and half new DNA, which is this pink right here. So you can also see here, like, this is a continuous line. So polymerase, when we're on this side, this is the 3' to 5' end, and I'll show you guys what that looks like here in a second. But all this is saying is that when you add new base pairs, it's easier to add to the 3' end. And in fact, that's the only way that we can do it. We have to add to a specific end of DNA. And so on this side, the 3' to 5' end, it's really easy for polymerase to just come in and just, like, you know, hook these guys on there and be like, woo, here we go. And so they're just, like, you know, moving right along. On this other side, though, the polymerase, what it has to do is add DNA to the 3' to 5' end. So what it will do is it will make these little fragments. They're called Okazaki fragments, and then it will attach them. And then the polymerase is going to skip up here and then build this direction, and then it's going to skip up again and build this direction, because each side of the DNA is opposite of one another. And so this side is the 3 or the 5 to 3' side. And so because of that, because of the way that we have to add those nucleotides on here, it's going to have to build in just little chunks, and then it's going to have to skip and then come up here and then build in little chunks, skip and then come up here and then build in little chunks. And then we've got ligase that will come in here and then attach these two together. So the discontinuous replication, so that's what I was just talking about here before with those Okazaki fragments. So DNA polymerase can only attach a free nucleotide to the 3' end of a DNA strand. And so, like I said, I'll if there's not a picture in this PowerPoint presentation, I'll show you what that looks like on Google. And so the DNA polymerase is going to attach a free nucleotide to the 3' end. That's the only place it can attach it to. And so only one of those two strands can be synthesized continuously, and those are the ones that go from the 3 to 5 prime. And so what we were looking at just before, that was on the right hand side, and so the polymerase there can just go ahead and be like, tick those things on there, like beep, 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 and just keep moving on up. The other strand, or the discontinuous strand, what it's going to have to do is make little tiny chunks of DNA and then skip up and then move down from 3 to 5 prime, skip up, move down 3 to 5 prime. And so it's going to have to do that in segments because it's the opposite direction. And so again, that side is going to take a little bit longer, and that side is where we're going to use our DNA ligase to join those little segments as we go along so that we can make one continuous strand of DNA. And again, those little fragments are called Okazaki fragments, and so you can see how to spell that right there, Okazaki, right there. And so again, this is the 3 prime to 5 prime. And so we have to build them this direction. That makes it so much easier. But if we have to go 5 to 3 prime, then we have to build them in like little segments like this. And then again, ligase is going to come in and it's going to attach those two segments together, and it's just going to keep doing that all the way up. And so this is the discontinuous end right here. And this is where that 3 and 5 prime comes from. So you can see on the sugar, so like this 5 carbon structure right here, the carbons are named for their position. So like here where we're attached to the nitrogenous base, this is carbon number 1, carbon number 2, carbon number 3, and so that's where that 3 prime comes from, carbon number 4, and then carbon number 5 out here. And so the 3 prime end is where we have to add new nucleotides, or adding them with the 3 prime end, and so that's why going on this side is easier. On this side, because the 3 prime end is pointing down, we have to do them in strips, and so we have to do them kind of backwards. Alright, so now we're going to talk a little bit about mutations, because sometimes these can be advantageous, but that's like the exception, not the rule. We don't really want mutations in our DNA. Every once in a while, like I said, a mutation can be advantageous, like blue eyes were a mutation. The ability to digest lactose was a mutation in the human genome a while back, and so sometimes these can be advantageous, but that's like I said, the exception, not the rule. And so because we don't want any mistakes made when we are making a copy of our DNA, we have polymerases that will come in and check the work of the other polymerases to make sure that the DNA is being copied correctly. And so these guys, their job is to proofread the DNA sequences and make sure that the DNA replication is going properly, and if they find a mistake, they can replicate it, or they can repair it, excuse me. And so we actually have repair mechanisms in place, and we have proofreaders in place, and we have several stops. And then if your DNA is doing something it's not supposed to be doing, sometimes the cell will just like trash everything it's doing and go through a process called apoptosis, where the cell just will program itself to die because it's not replicating properly. So there are some things that can cause DNA damage. Some of those are what are called ionizing radiation, because they turn your DNA into like ions, and so it knocks electrons out of your atoms, and this will make how the atoms are going to be attracted to or repel things that are of positive or negative charge, and so it'll fold, it'll do all kinds of weird stuff. And so gamma rays, X-rays, UV light, all of those things can damage your DNA. This is why when you go get an X-ray, if you're getting an X-ray of your leg or something, they come in with like the giant lead cape and put it over your innards and put it over everything that's not getting X-rayed. And they only X-ray the part that needs to be X-rayed. And this is also why the little nurse or the little X-ray technician that comes in, and she puts everything on you and puts the big lead cape on you or whatever, she runs behind the wall, right? And then she'll push the button and then she comes back out. Because X-rays can damage your DNA and they can cause cancer. So it can break up your chromosomes into pieces, and those pieces might get lost. It can create free radicals in your tissues, really not good. And then UV light causes what's called a thymidine or a pyrimidine dimer that can kink up your DNA strands. This is not good. This causes skin cancer. This is why going to the tanning bed is such a bad idea. If you know, a little bit of sunlight is good because you need to go out in the sun so you can produce your vitamin D. But if you're in the sun a lot, you need to wear your sunscreen because UV radiation will cause skin cancer. It's bad. It causes those pyrimidine dimers, and then it causes your DNA to get read incorrectly. So there are some chemical carcinogens, the cancer-causing agents out in the environment, and so like tobacco smoke, I think we all know at this point tobacco smoke literally causes lung cancer. There are 55 carcinogenic or cancer-causing chemicals in tobacco smoke, and so don't smoke. I mean, oh my God, like don't smoke, so bad. Don't go to tanning beds. Do not smoke, and wear your sunscreen if you're going to be out in the sun a lot, okay? And smoke is a carcinogen of itself, any kind of smoke. So if you're like a barbecue person, and you go outside to your barbecue pit and then like inhale deeply for 5 minutes 20 times a day, that can cause lung cancer too. So all smoke is carcinogenic, but tobacco smoke is like especially super fun and carcinogenic because of the way that tobacco is grown. It's sprayed with all kinds of pesticides, it's treated with all kinds of crap, and then not only that, you roll it up, get it really close to your lungs, and then you light it on fire and then breathe it into your lungs. And so, I mean, all that tissue that's in there is so sensitive. And so even just like being hot from the smoke can cause all of the epithelial tissue in your throat and in your trachea to turn into stratified squamous epithelia, which is bad because you want it to be like that simple squamous with the simple pseudostratified ciliated columnar epithelia. Oh my God, that was like such a huge term. But you actually change the tissue in your throat just from the heat exposure from one type to another. And then also you get all those carcinogens in your little alveolar sacs or whatever, so just don't smoke, okay? And also do not go out to your barbecue pit and inhale deeply 20 times a day. Don't inhale smoke if you can help it. There are lots of other environmental pollutants, like there's endocrine disruptors, there are carcinogens, all kinds of stuff out in nature and also just out because of our industrial kind of nation. And all these chemicals and things are really not good for you, and some of them can even irreversibly bind to your DNA, and those cause replication errors, and then that leads to mutation, and that leads to cancer. So not good stuff. Rosalind Franklin, if you guys remember, she did the X-ray crystallography, and she's the one that did all the pictures and stuff for Watson and Crick. She actually died of cancer at 37, okay? And it's probably because she was exposed to so much X-ray radiation. Because way back when, they didn't really know that X-ray radiation, they were just figuring out what DNA was. Like, they didn't know that X-rays caused like ionizing damage to your DNA. They were just figuring out what DNA was. And so she was exposed to all this X-ray radiation, and so she got cancer and died, and it was probably very, very closely related to the fact that she was always exposed to these X-rays all the time. So not the best thing, and she was really brilliant, and she died at 37. I mean, she wasn't even 40 yet. She was young. So one of the other fun things that we can do with DNA is we can clone things. And so cloning means that you make an exact copy of a molecule, a cell, or an individual. And so this can occur in nature with asexual reproduction. So when you've got like a planarian worm, for example, you can chop them in half, and each half will turn into a clonally identical adult. So basically what you've got is two individuals that are exactly the same. So cloning means you've got an exact copy of an individual. So what we have to do with cloning is kind of special. So as your cells develop, they become what's called differentiated, which means that they become kind of solidified in their job. Like a bone cell, for example, is never going to turn into a liver cell. It's differentiated. Okay, it's got its job, it knows what it's doing, it's never going to become something else. And so that's what they mean by differentiated, is they're different now and that they're not going to turn back into something else. So they're different in form and function, and this is generally a one-way process in animal cells. Some cells and some animals can reverse this, and they can go back to a stem cell state, and they can regrow their tissues, like planarian worms, for example, can do this. So reproductive cloning means that we're going to produce an exact copy of an individual. I'm almost done, Bobby, okay. So what's called somatic cell nuclear transfer, or SCANT, that's what I call it, SCANT, is nuclear DNA in an adult that is differentiated. Okay, we can transfer that into an enucleated egg. And so the egg cytoplasm, so all of the ribosomes, all of the different organelles and things that are going to help us produce our proteins, they can actually reprogram that differentiated, that already jobified DNA to act like an undifferentiated DNA. And undifferentiated is a stem cell, okay? So undifferentiated means that it doesn't have a specific job yet. So embryonic stem cells, for example, like you started off as one cell, And then from that one cell, you became a skeleton, you became a muscular system, you became a digestive system, you became all these different things, but all that came from one cell. That one cell at that point of conception was undifferentiated. It did not have a specific job yet. And so we can actually reprogram our adult DNA to act like stem cell DNA, which is kind of cool. And so this is how they do that in a petri dish. And so you've got your egg cell right here, and then we've got our DNA from an adult. So we can like stab that little eggy guy right there. We're going to poke in our DNA. We're going to leave it in there. And then we can actually get those differentiated DNA, like adult DNA samples, to turn into stem cells. And so from there, we can do things like therapeutic cloning. And so this is where we can produce human embryos for research purposes. We can harvest undifferentiated or stem cells from those cloned human embryos, and then stem cell research can take place, which is kind of interesting, and it's actually like a really, really awesome thing. So the whole point of stem cell research, and the reason that people get so excited about it, is because we can actually look for treatments for people who suffer from fatal diseases, because you could actually reprogram their tissues to act like they were never sick in the first place, which is really, really cool. And so this is like up and coming. Kind of difficult to do that in the United States because there are ethical problems still with them using embryonic stem cells and things like that. But in Europe and in other places where some of those sanctions have been lifted, people can do a lot of research, and they can do a lot of interesting, like really cool things with stem cell research. So that's it for Chapter 8. These are some points to ponder, so look over those as you're doing your reading and stuff this week, and I will see you guys later.